Welcome to the Everyday Journal Club. I'm Hope, a biologist with 15 years of research experience, now fortunate to be working in a Nobel Prize-winning lab. This channel has three main goals. 1. To help you stay effortlessly updated on the latest scientific breakthroughs. No paywalls, no jargon overload. 2. To build a supportive community where we can exchange the most valuable experience-based research advice. 3. To share valuable strategies and concrete examples on how to achieve meaningful scientific breakthroughs. Once we reach 1,000 subscribers, I'll begin releasing content focused on the third goal. Let's go! Let's make great science together.
Okay, let's unpack this. Imagine you're holding a treasure map, but it's not a drawing. It's a string of invisible letters, billions of them, encoding everything about a living organism. For decades, reading that blueprint DNA was like trying to decipher it in the dark with fragmented clues. Then, in 1977, a groundbreaking paper dropped, introducing a method so simple, yet so profoundly clever, it finally handed us a clear alphabet reader for DNA, utterly transforming biology.
And transforming isn't an overstatement, really. What's truly revolutionary here is how they looked at a basic biological process, DNA replication, and found a way to hijack it. They leveraged a common enzyme, DNA polymerase, and a surprisingly simple dead-end molecule to achieve what complex, hazardous chemical methods couldn't. A scalable, accurate, and, while truly elegant way to spell out the genetic code base by base. It was less an improvement and more a fundamental rethinking of the problem, you know.
You're right, it feels like a genuine eureka moment. Before this deep dive, reading DNA was a bit like trying to solve a puzzle, with half the pieces missing and the other half covered in, well, toxic goo. Early methods were either too short in their reads, too prone to errors, or involved harsh, complex chemistry that made high throughput practically impossible.
Exactly. Take Sanger's own earlier plus and minus method. It was rapid, sure, but the results could often be ambiguous, demanding multiple confirmations. Kind of frustrating. Then there's the Maxim Gilbert chemical degradation, which had its strengths. It could work on double-stranded DNA, but it was incredibly laborious, relied on hazardous chemicals, not exactly user-friendly for routine lab work. What the scientific community wasn't just looking for was a better method. They were desperate for a universally usable, high-accuracy approach for determining long DNA sequences. Something simple, reliable, and scalable for, every lab, not just a few specialized ones.
So the stage was really set for a massive breakthrough. The urgency was palpable, with a race kind of brewing to decode genetic information. And the small, easily manageable genome of bacteriophage sacs 170 and 4 was the perfect proving ground for anyone who could crack the code better. The scientific world was truly holding its breath for that aha moment. Okay, so knowing the problems with older methods, what was the aha moment? How did they flip the script from hazardous chemicals to something far more elegant and biological? I'm picturing some ingenious biological hack that sidesteps all that harsh chemistry.
You've pretty much hit on it. The core idea was brilliantly biological, co-opting DNA polymerase, the enzyme that naturally builds DNA, and then introducing special dead-end building blocks into the reaction. The brilliance here lies in a tiny chemical alteration. These 2'3' dideoxynucleotide triphosphates, or DDNTPs, lack a 3' hydroxyl group. Now, this isn't just a structural detail, it's the master key to their function. It means once incorporated into a growing DNA strand, that strand hits an irreversible dead end. No more bases can be added. This simple chemical trick allowed them to precisely control where replication stopped, turning a complex biological process into a readable sequential output.
So it's almost like a bricklayer, occasionally using a special flat-top brick that physically cannot have another brick placed on top of it. You end up with a wall where the bricks stop at various points where that dead-end brick was used. It creates a kind of molecular ladder of different lengths, each telling you something important.
Precisely. That's a great analogy. To read a sequence, the method involves setting up 4 parallel reactions. Each reaction contains a primer, the DNA template you want to sequence, the DNA polymerase, all 4 normal deoxynucleotide triphosphates, DNTP, your standard A, C, G, and T building blocks, and crucially, a small amount of one specific DDNTP. So one tube gets DDDATP, another gets DDCTP, another DDGTP, and the last gets DDTTP.
Right, and to make this invisible ladder visible, one of those normal DNTPs in every tube is radioactively labeled, usually with 32P, so we can actually see these newly synthesized DNA fragments that are being created. Is that how you track them?
Exactly. That's how you visualize it. In each tube, the polymerase extends the primer, but every so often, just by chance, it incorporates the DDNTP specific to that tube. When that happens, boom, the chain terminates specifically at that base. This process, repeated millions of times in the tube, produces a unique set of fragments of varying lengths. All these fragments start from the same primer, but each ends at every instance of the specific base, A, C, G, or T for that particular reaction. It's essentially random yet totally controlled termination.
Okay, so we've got these 4 distinct sets of terminated fragments, each a snapshot of where an A, C, G, or T was. But
Yeah.
how do you piece that together into a continuous sequence? That seems like the next big hurdle to visualize.
That's where the trusty denaturing acrylamide gel electrophoresis comes in. You basically separate these fragments by size. The shorter fragments, being lighter, zip faster through the gel matrix, and the longer ones move slower. It's like a molecular race. When you run all 4 reactions side by side in separate lanes on the same gel, you get this distinct pattern of bands. By reading these bands from the bottom of the gel, which represents the smallest fragments, the bases closest to your primer upwards to the largest fragments, you can directly spell out the DNA sequence, base by base. It's literally an alphabet emerging before your eyes on the film, a direct readout of the genetic code. Quite beautiful, actually.
That is quite elegant. Let's see how they demonstrated this in practice. Figure 1 of the paper, for instance, beautifully illustrates the core elegance of the method, right? Showing how they achieved robust sequencing even in its simplest form.
Absolutely. Yeah, figure 1 is a great starting point. Here, small restriction enzyme fragments, A12 and A14, were used as primers on the complementary strand of Hevex 174 DNA. The beauty of this initial setup was its sheer simplicity. There was no need for a complex final digestion step to cleave the primer away from the newly synthesized DNA before running it on the gel. You just ran the reaction product directly. Super straightforward.
And the paper mentions they used not just DDGTP, DD-ADT, ATP, DDDP, but also an arabinonucleoside called ARA-CTP as an inhibitor. So it wasn't just dideoxys that could achieve this stop. They were casting a wide net for what worked.
Correct. Yeah, they were exploring. They also found that arabinonucleosides like ARA-CTP could function as chain terminators. This showed their ingenuity in exploring various chemical forms that could achieve that critical stop, demonstrating that the principle of chain termination was more important than the exact molecule. Smart thinking. In this simple setup, they could read about 80 nucleotides, starting 88 nucleotides from the 5-foot end of the primer. Now, there could be some difficulty resolving consecutive identical bases, what they called runs. Sometimes they compress a bit on the gel. But the overall pattern was remarkably clear, a significant improvement over previous methods.
80 nucleotides is great for a start, but in biology, we often need to read much longer stretches of DNA. This next adaptation, shown in Figure 2, was crucial then, right? For moving beyond short reads, demonstrating the method's scalability and, impressively, its ability to correct errors in existing genomic maps.
Precisely. Yeah, Figure 2 tackles longer reads. With longer restriction enzyme fragments, like fragment R4 used here, the primer itself is longer. So, to accurately read the newly synthesized sequence without interference from the primer's length contributing to the total size, it becomes necessary to split the primer off from the new DNA chain before electrophoresis. This was typically done using another restriction enzyme, like He3, in this specific experiment, which cuts at a known site between the primer and the newly synthesized bit.
Did this longer read approach introduce any new quirks or challenges into the process? You're adding another enzymatic step, which always has the potential for complications.
It did, yeah. Good point. Interestingly, for these longer fragment experiments, they found that only the dideoxynucleotide triphosphates worked well as inhibitors. That ARA-CTP gave less satisfactory results when restriction enzymes were involved in this splitting step. Now, this might be due to the DNA polymerase's 3' exonuclease activity. Its proofreading function may be cheering back the ARA-C at the higher temperatures needed for the restriction enzyme digestion, or perhaps even the enzyme managing to extend beyond ARA-C under those conditions. It highlighted the importance of picking the right stop molecule for this specific experimental setup.
But the payoff is huge, right? This experiment yielded a 120 nucleotide sequence and significantly helped correct a provisional error in the published TVEX 174 genome, identifying a missing CGC trinucleotide. That's not just sequencing, that's validating and improving our understanding of an actual genome.
Absolutely. That's a huge win for accuracy and shows its immediate utility in validating existing data. It wasn't just theoretical. But this deep dive also raises an important point, maybe something for you listening to consider. Even with this elegant solution, what happens when the DNA sequence itself creates problems on the gel? They noted these pileups where multiple bands appeared at the same or very close positions. These are often caused by the DNA forming stable, base-paired loops, essentially, the DNA strand folding back on itself like a knot, which messes with how it moves through the gel. For instance, in Figure 2 at position 4330, they saw 4G residues appearing as a single strong band, likely because they were pairing with 4Cs nearby in a hairpin loop structure.
It sounds like even with this elegant solution, DNA itself could still be a bit uncooperative. You're trying to read a precise message and starts folding itself into origami. Was that a common headache in the early days? And speaking of tricky situations, what if a restriction enzyme site needed for that splitting step was too close to another one, making a clean cut for primer removal impossible? How did they creatively solve that problem?
That's a great observation. And yes, secondary structures, those loops and folds, were definitely a common headache. Still can be, sometimes. For your second question about the restriction site issue, that's where the single-site ribosubstitution method, shown in Figure 3, offered a really elegant workaround. Instead of relying on a restriction enzyme cut, they incorporated a single ribonucleotide, an RNA building block, basically into the DNA primer itself during synthesis. Then, after extending the sequence with the inhibitors, the primer could be precisely split off using ribonuclease or alkali treatment, which specifically targets and breaks the RNA link, leaving the newly synthesized DNA intact. Clever, huh?
Very clever. And this was particularly useful for restriction enzymes like ALU-I, which the paper notes are inhibited by single-stranded DNA. So they couldn't be used for the usual splitting method if the template wasn't fully double-stranded nearby. It's a smart way to bypass those enzyme limitations.
Exactly, it gave them flexibility. Figure 3, using fragment A8 on the viral strand of FEX174, allowed them to read about 110 nucleotides using this ribosplitting trick. Crucially, it led to another significant revision of the provisional FEX174 sequence. They corrected a segment at positions 3524, 3530 from ATTCAC to ATCAAC. Another correction found. And they also encountered a similar pile-up issue here, suggesting another loop structure, which they later managed to resolve, finding an extra C by running the gel at a higher temperature to help melt the loop. It really shows how inventive they had to be, tweaking conditions to get reliable reads in the face of these inherent challenges with DNA behaving badly on gels.
But if we pull back and look at the big picture, what does this all mean for the field of biology? This new method, Sanger sequencing as we call it now, wasn't just an incremental step forward, was it? It sounds like it fundamentally reshaped how scientists even conceived of sequencing, wouldn't you say?
Absolutely, there's no question. Compared to Sanger's earlier plus and minuses method, it was fundamentally simpler to perform. Fewer steps, fewer incubations, less purification needed. It utilized the readily available Klenow fragment of DNA polymerase first, which was a workhorse enzyme in many labs already. And critically, it produced clearer results with fewer artifact bands, allowing for significantly longer reads, typically 15 to 200 nucleotides, sometimes even pushing up to 300 from a single primer run. This was just an enormous leap in practical utility and read length compared to what came before.
And it solved that annoying problem of estimating links in those runs of identical bases, right? Where multiple A's or T's might blur together. With Sanger sequencing, each base in a run generally showed up as a distinct band, making it much more accurate and less ambiguous.
Precisely. That was a major source of errors in earlier methods, and this method largely eliminated it by resolving individual bases within runs. Big improvement for accuracy. Moreover, its efficiency with radioactive labeling meant it could be performed on a smaller scale. You didn't need vast amounts of material. This really democratized access to sequencing for many labs that didn't have the resources for the more complex chemical methods. It brought DNA sequencing out of specialized centers and into everyday molecular biology research.
So this wasn't just an incremental improvement. It was a theoretical and technological reframe. It demonstrated that sequencing could be tackled as an enzymatic termination problem, shifting away completely from that hazardous chemical cleavage paradigm. This innovative mechanism of using ddNTPs as precision stops set a completely new standard.
Exactly. It moved us away from brute force chemical degradation to an elegant enzymatic solution. That fundamentally changed the research landscape. This method, Sanger sequencing, quickly became the dominant platform for, well, decades, really. It was the technology that gave us the first complete DNA genome, that same X174 phage they used as the test case in the paper. And it powered the early foundational stages of the Human Genome Project. The impact is truly hard to overstate. It laid the groundwork for everything that followed.
Wow. Its applications were immediate and vast, weren't they? From routine verification in gene cloning and mapping specific genes, to sequencing entire viral genomes and eventually discovering disease-causing genes in humans. It truly set the stage for modern genomics as we know it. making DNA sequencing accessible and reliable for the first time on a really broad scale.
But even for such a groundbreaking method, which seems so robust in principle, there were still some early limitations they had to contend with, weren't there? Nothing is perfect right out of the gate.
Certainly. Yeah, you have to remember the context. Early implementations still required radioactive labeling, which has safety and disposal issues, and manual pouring and reading of gels, which inherently limited throughput. You could only run so many samples a day. The pileup issue from secondary structures could cause local errors if you weren't careful or didn't try different gel conditions. And while read lengths were impressive for the time, they weren't yet suitable for tackling truly massive genomes like the human genome in a single go. You had to piece together lots of smaller reads. Also, just practically, getting all the necessary DDNTP inhibitors, especially DDGTP, could apparently be difficult sometimes, as they weren't always reliably commercially available right at the outset.
Right. So the advice even then was clear, and it's still good advice today. For true accuracy, don't rely solely on one run or one method if you can help it. Confirmation, maybe from sequencing the opposite strand or using a different primer, was always good practice. That critical thinking approach, cross-referencing your data, remains vital in any scientific endeavor.
Absolutely. This work, born in Fred Sanger's lab, known for its method-driven biology and collaborative spirit, provided a practical, modular workflow that truly transformed how we understand and manipulate life itself. It just shows the power of simple, elegant biological solutions to complex problems, doesn't it? And how a breakthrough isn't always about the biggest, most complex machine.
It's incredible to think that the fundamental logic converting a DNA sequence into a series of discrete, rootable signals using these precision stops, that core idea still underpins so many of the advanced, high-throughput sequencing technologies we use today. Different chemistry, different detection, but the same basic principle. It's a real testament to the power of a simple, elegant idea that revolutionized a field and continues to echo in our modern genomics.
It really makes you wonder, and this is maybe something for you to consider, what other fundamental biological processes might hold the key to unlocking the next generation of biotechnological breakthroughs? What other processes are just waiting for us to find their precision stop?
Something for you to ponder as you look at the cutting edge. We hope this deep dive into Sanger's seminal 1977 paper has given you a newfound appreciation for the ingenuity that built the very foundation of modern genomics.
Absolutely. Until next time, keep exploring.